Hello, and welcome back to our series of lectures on action potentials. In this little section, we're going to be talking about the phases of the action potential. And we're going to be answering these questions in this little segment here. One, what are the phases of the action potential? And two, what events underlie each phase? Okay, so remember that action potentials have a stereotyped shape. They're all or none. They're always going to look the same every time you have an action potential in the same neuron. Okay, they do vary a little bit from neuron to neuron, but within the same neuron, the action potentials are always going to look the same. Taking a look at this figure here, what we're looking at is we are recording the membrane potential of this neuron at one location. So I want to just kind of point out that that's what we're looking at here. We're looking at this signal, right? So this action potential that's happening over time at this one location. So again, that's a very stereotyped set of events, and we're going to be looking at that. We have names for those stereotyped, or for the parts of that stereotyped shape. There are stereotyped events that happen that lead to that stereotyped shape. We're going to be talking about all of that in this section, okay? All right, so first off, let's start right here where it says number 1. The neuron is going to start at its resting membrane potential. And I just want to just kind of start off by reviewing, what does the membrane potential look like? Why is it at that value? So take a moment, hit pause, answer that question on your worksheet. Okay, so I want to remind you, not too hard to figure out by looking at the graph. Typical resting membrane potential in a neuron is negative 70 millivolts. Hopefully you remember why that is, right? So the main reasons for that are, 1, we have a lot of open potassium channels. We have a lot of potassium leak channels present. We have some open sodium channels present, so some sodium leak channels. Since we have so many potassium leak channels present, potassium is going to be able to move towards equilibrium. It has a big opportunity to move towards its equilibrium. Remember that its equilibrium potential is negative 90. So potassium is going to move in such a way as to bring the membrane potential down towards negative 90 millivolts. However, it's not able to bring the membrane potential all the way down to negative 90 millivolts, because we do have those sodium leak channels that are present. And so sodium is going to be able to move a bit towards its equilibrium potential of plus 60 millivolts, okay? Just want to remind you, sodium concentration outside of the cell is very high. Potassium concentration inside of the cell is very high, and that concentration gradient is set up and maintained by the sodium potassium ATPase. All right, so that's the resting membrane potential. The start of an action potential, the action potential is initiated, I guess, by a depolarizing stimulus that comes in towards the axon. Just incidentally, the depolarizing stimulus is actually a graded potential that's coming from the cell body, but we'll talk more about that later. For the time being, just keep in mind, we have something that's coming in and depolarizing the membrane potential right here, okay? When the membrane potential depolarizes, if the stimulus reaches threshold, an action potential is triggered, right? So we have this value right here called threshold, which is usually about negative 55 millivolts. If the membrane potential is depolarized to that level, then we'll have an action potential. Okay, and here's a question for you. What do you think will happen if the stimulus does not reach threshold? Go ahead and answer that question. Hit pause if you need to. Okay, so if the stimulus does not reach threshold, then you won't get an action potential, right? So again, remember that it's all or none. So this is like sort of that threshold for the action potential, right? So if you hit this threshold, then it's all. That's the whole shebang, the whole action potential. If you don't get there, nothing. You don't get like a miniature action potential or anything like that. Okay. All right, so moving on. Once we depolarize the membrane to threshold, that triggers the opening of a whole bunch of sodium channels. Okay, so these sodium channels, which we'll talk about a little more in a little bit, these sodium channels are voltage gated. So if we reach this voltage, then those channels will open. And so we're going to have a sudden increase in sodium permeability. Okay. And so here are some questions for you. What's going to happen when sodium permeability increases? And secondly, in what direction will sodium ions move? So take a moment to answer those questions on your worksheet. Okay, so let's go over the answers. When sodium permeability increases, sodium's suddenly going to have a really big opportunity to start moving towards its equilibrium, right? So remember that sodium concentration is very high outside of the cell. Sodium equilibrium potential is plus 60 millivolts. If we are starting at negative 55 millivolts and sodium wants to be at plus 60 millivolts, it's going to move in such a way as to bring the membrane potential closer to 60 millivolts, now that it, closer to positive 60 millivolts, now that it has the opportunity. And that's going to cause the membrane to depolarize. Sodium ions are going to need to move into the cell in order to do that because they're positively charged. When we move into the cell, that will make the inside of the cell more positively charged, or in other words, it will bring up the membrane potential. Okay. All right, so we kind of already talked about this, right? So we opened up a whole bunch of sodium channels. Sodium can come into the cell. Lots of sodium comes into the cell because we have suddenly opened up a lot of sodium channels. And that's going to cause the cell to rapidly depolarize. We have a name for this part of the action potential. It's called the rising phase or the depolarization phase.